I'm Mr. Wilson from the cover page, and today we'll be looking at microbes and some keywords used on the exam paper. I hope you're all well and ready to learn. We put out a video every day, so remember to like, share, subscribe, click that notification bell, and most of all, leave us a comment. Now, let's go. So today, as stated, we will be looking at microbes. Objectives of today: draw and compare structure of a generalized bacterial cell to the plant cell and animal cell. So we're going to be comparing the plant cell and animal cell to the bacterial cell. We want to also draw and examine an amoeba cell. We want to identify economical importance of microbes, and we want to look at some keywords used in the exam. Alright, so these words in yellow, these are some words we consider to be keywords, and we are going to be defining them, exploring them a little bit in the paper to come. So as we go along, you'll see us popping out a word and doing the explanation. Then we get into some questions. So compare. Compare is to note similarity and differences, or to look at similarity and dissimilarity between things, place, and you could name it. Just about anything that you could look at their similarity and their differences. You could probably look at a boy and a girl. Alright, you could look at probably two cars, what makes them similar, what makes them dissimilar. You could compare just about everything that you can see around you. It is a skill that you need to develop. Understand the term compare so that when you see it on the exam paper, you're able to use it well. You must be able to use the word examine, have an appreciation for the word. To examine is to inspect or to look at person or thing thoroughly in order to determine their nature, makeup, functionality, or condition. Now, we all can speak to the fact of a doctor examining us. You can speak to a mechanic looking at your vehicle, examine. The word examine, we're seeing it so many times around us, or so many times we've seen person employing this skill to examine, to inspect, to look at something thoroughly, and then they come up with some a formula, some idea, a prognosis. Something comes up that they can use to move forward. You're feeling sick, the doctor would examine you to say if you are probably functioning well, if everything is alright. So examine, you want to be sure that you can use that word properly and have an appreciation for it in your exams. Explain. Is there a day that you haven't explained anything? To reveal relevant facts which bring greater clarity to a subject. And I might just need to explain here. Subject, just about anything that you're talking about here would have been referred to as the subject. So to explain, you want to reveal relevant facts. Don't bring up things that are not relevant. Relevant facts which bring greater clarity to the subject. So you're doing science, ensure that the explanation that you're providing is rooted in scientific facts. Now, microbes. What are microbes really? Microbes are small organisms, most of which are responsible for disease. I think that is the first thing that is going to be popping out from anyone. What are microbes? Something that causes disease. Now, except for a few fungi that are a part of this group, all the other belongs to the kingdom prokaryote and protoctista. Alright? Now, it is important to understand that microbes because they have economical importance. And when we talk about economical importance here, let me do some explanation. So I'm explaining. Economical importance does not mean that you're going to sell them. What it means is that they could impact you positively or negatively, either costing you money or bringing money to you. All right, so you might be able to gain from them or you might be able to lose from them. And it's not always money when we speak about economic in this way. All right, you might get healed, and of course if you get healed, the family might be losing some money. So it's very, very important that we understand the economical importance of microbes. And then the other thing we need to take in consideration is that microbes are not all bad. Now, microbes are found in the nitrogen cycle. You could talk about Nitrobacter, Nitrosomonas, and you could go with a whole long list of other useful bacteria. Now, you could look at recycling of nutrients. We have seen that we could look at the many diseases that are caused by bacteria, and of course the spoilage of products which are caused by other pathogen, other microbes. Now, the farmers will complain bitterly about microbes and their crops. The persons who are manufacturing in the food manufacturing process, they would have a wonderful or a great appreciation for microbes, because microbes could cost them a whole lot of money. Microbes could actually cost them their business. So it's very important that we understand microbes and we don't treat this topic with scant regard, because microbes, they are everywhere. In a matter of fact, microbes tend to live in conditions that humans couldn't even dare think about to be a part of conditions like those. Let's continue. So we want to look at the bacteria. Bacteria, bacterium. I want you to tell me in the comments, what's the difference? So bacteria are single-celled prokaryotic organisms that are found almost everywhere in the environment. Some live in very extreme conditions of temperature and pH. Now, bacteria, they move by what we call a flagellum. It's something like a tail at the end. If you have ever seen a diagram of the sperms, it's something that they pretty much whip in the water, whipped in whatever medium they are in order to move, right? So if you say the sperm swim, then it's probably nice to associate the bacteria movement to that. But it whips that tail, that flagellum, in order to move. Now, there's something I want to point out. The fact that I want to look at the word prokaryote or prokaryotic, and I want to ensure that we have a good grasp of it. I have brought it in a lesson before. I'm having it here again, and I'm going to be having it again. So we spoke about prokaryote, and we also spoke about eukaryote. Now, you don't need to study both words, but you need to have an appreciation of both. Now, the eukaryotic organism, if I were in your position, I would think about just that one. When we talk about eukaryotic organism, we are talking about our cells. We have membrane, we have the nucleus that is bounded by a membrane. So if I'm saying eukaryotic organism, yes, I am one of those organisms because my nucleus is bounded by a membrane. So if I am not talking about eukaryotic organism and I'm talking about prokaryotic organism, then I am talking about organisms that have nucleus that is not bounded by a membrane. So I am not a part of the prokaryotic group. I am a part of the eukaryotic. So remember, eukaryotic, yes, you are a part. Prokaryotic, you are not a part. All right, let's continue. Now, bacteria, they are not all harmful. Some are useful and even live in the gut of animals and help with nitrogen fixation in plants. Now, this word is going to come up when you are looking at the nitrogen cycle, the Nitrosomonas bacteria. All right, so you want to underline that one. You want to jot it down somewhere. You want to go and get a little more reading on it because it is going to pop up at you again. Now, bacteria are also used for medicinal purposes. Bacteria, they are also used for medicinal purposes. I want you to jot down that for me, and I want you to write in the comment section one medicinal purpose that you know bacteria is used for. All right, in industries, Did you know that bacteria is used in the production of cheese, soy sauce, vinegar, yogurt, and the list continues, and it could go on and it could go on and it could go on and on and on and on. But bacteria, they are used in industries. And I know everybody like the bun and cheese, and everybody have a little vinegar on the vegetable, and every now and then you get a little yogurt and so on. So I'm sure that everybody can relate to these products. Yes, if you know anything about them, there are some persons who are allergic to these products. But if you know these products, bacteria was a part of the manufacturing process. All right? Good. Let's move on. It is important to note that most are harmful. Yes, most bacteria, they are harmful, but we still have some good ones, and we really don't want to leave that bit out. All right, so most bacteria are harmful, and we can make reference to this one, which might come later in the syllabus when we are looking at health. The gonococcus bacteria, and the gonococcus bacteria, that bacteria is responsible for gonorrhea. You might hear about gonorrhea. It is responsible for gonorrhea. And for those agri students who will be looking at zoonosis, yes, gonorrhea is a zoonosis. It's a disease that can be transferred from animal to man. Gonorrhea is usually seen in dogs and all of that, and it can transfer from, of course, animal to man. So we refer to it as a zoonosis. Don't want to touch that too much now. We're not doing agri, we're looking at bio, and then later we'll be looking at HSB. So this is what the generalized cell of a bacteria look like. I want you to pay close attention to the screen. If you observe here, right, you are not seeing any nucleus that is bounded by a membrane, right? There is no nucleus that is bounded by a membrane. Let us look at the parts. So that little area there you're seeing first, we have the plasmid, which is a small piece of DNA, and you would have an appreciation that DNA is what bears our genetic information, or bears the genetic information for this organism. Remember, we said earlier in some video that an organism refers to any living thing. Beautiful. Capsule. Now, this area is a slimy covering that protect the bacteria, and it would be found pretty much on most bacteria. All right, most bacteria. So we have bacteria, bacterium. Help me there. All right, so this capsule, this is a slimy covering that protect the bacteria. Now, cell wall. I have some words here in colored green. And these words are in this color because we know are comparing. All right, so comparing, contrasting. So you'd remember that the plant cell has a cell wall, but the plant cell does not have plasmid. The plant cell does not have a capsule. Let us continue. Cell membrane. The plant cell has a cell membrane. The animal cell has a cell membrane. And here too, the generalized bacterial cell is showing a membrane. And the function, they are all outlined there in white. I want you to look at them carefully. I'm going pretty slow because I want you to have an appreciation for what we're doing. And at any time throughout this video, feel free to pause, feel free to go back and start watching from the start. Go over the video as many times as you wish, so as to ensure that you understand the content that has been presented. Now, if you still don't understand, leave us a comment and we'll follow up in another video. All right, cytoplasm. Who can forget cytoplasm? It's the inner portion of a cell where the organelles are suspended. Now, I'm going down to some fancy thing I wanted to talk about. Nucleoid. When we started out, we were looking at this area, and we said that there was no nucleus that was bounded by a membrane, right? So the nucleoid, it's pretty much a coil of DNA. It's like a strand, like you have some thread for those persons who understand that bit, and it's all coiled, right? pretty much it's like DNA all wrapped up, wound up, and it's not bounded by any membrane. Now, if you want to know what makes a big difference here between the prokaryotic cell and the eukaryotic cell, then this is where you're going to be looking at, because here you're going to realize that while in your cell, an animal cell, a plant cell, you will have the nucleus all encapsulated in a membrane. Here in the bacteria, there is no membrane that forms an envelope or an envelope for the nucleoid. And remember, the nucleoid is what is really carrying most of your genetic information for this organism. Let's move on. Ribosomes, we're going to be looking at that we're going to be looking at that when we look at HSB. But the ribosomes, they are pretty much found on the endoplasmic reticulum. We have not looked at that yet. We have the rough ER and the smooth ER, and we're going to be looking more at that in other lessons. So don't get too anxious about it. Yes, we did not look at it when we were looking at cells, but if you up on over to the HSB, the Human and Social Biology lesson, you'll see us talking about the ribosomes. Now look at the flagellum. Interesting here, that little tail-like structure at the end of the bacteria, pretty much it allows the bacteria to move along. So it gives us whip-like movement moving along. And remember, most organisms can move, and never for a minute confine yourself that movement is only about swimming, walking, running, flying. We're going to come up on some other types, some other ways in which organisms move. I'm sure you'll be fascinated. Now, we could think about organisms that move pretty much by whipping what we call cilia, but we're not going to be touching that yet. Later in the lesson, we're going to be looking at that. So I do hope you have a grasp of what is on the screen. You're able to use this color-coded word to look at the similarities and differences between animal cells, plant cells, and bacteria. All right, so from at any point, feel free to up on over to the other videos. I'm going to be putting a link in the description below this video, so ensure that you watch and watch and watch and watch, and just play around with these videos. It's COVID time, you're all locked in. I'm going to be putting some nice soundtrack to the end of this video, so you should be well taken care of and be enjoying watching and learning and listening to music at the same time. Leave me a comment and let me know how you found that bit of a music combination with the lesson. Let's look at the amoeba. Now, this is going to be new to a lot of persons, but fear not. This is going to be new to a lot of persons, but fear not. There is a link in the description below, and you'll see what the amoeba look like. You'll see how it moves, what we call cytoplasmic streaming. You'll be seeing that later in the content. So as soon as you're finished with this video, ensure that you go down to the description and you're going to be looking at the supporting videos for this content. All right, the amoeba. The amoeba is a member of the Protoctis kingdom. They move by cytoplasmic streaming with what we call pseudopodia. Pseudopodia, false leg. Pseudo, false leg. For those persons doing agri, banana is not actually a tree. It is a pseudo tree. It's a false plant. So pseudo, false. So pseudopodia, the amoeba moves by what we call cytoplasmic streaming using what we call false leg, because it can just move that membrane about and get it to move. I mean, watching that video, it's going to be a real wonderful video. So ensure that you, when you're finished watching this video, you up and down to the description, click on the link, and you will go right into a video that takes you to the amoeba. Now, these may be found in some water bodies. Amoeba might be found in some water bodies. Interesting. They can also be found in the human gut, causing what we call dysentery. Now, dysentery is running belly, and for most persons, when they have a little runny belly, it comes with a little fever and a little pain. So then you see that there's economical importance here, because when you get sick, you can't go to school, your parents can't go to work, and then we are losing out on some income, the bills have to be paid and all of that. So we talk about economical importance. Once you get sick, now you know what happens thereafter. They are parasitic. parasitic and care and are of medical importance. Parasitic, big word coming out there, big word, parasitic. All right, so parasites usually live on their hosts and they cause them harm. And another thing we know is that parasites are usually from different species. So never you go around saying that a baby is parasitic to its mom. No, they are of the same species. So a parasitic organism usually feed on its host and usually cause it harm. Right? Remember, if something is dead, you can't cause it any harm. All right? So we want to be careful. This is a nice word. I want you to keep it because we're going to be looking at it later in some other lesson. Let's move on. Now, the amoeba cell, look at this. Awesome. The thing about it, you might be seeing many different diagrams of the amoeba. Many different diagrams. Because like I said, the amoeba move by what we call cytoplasmic streaming. And because it moves by cytoplasmic streaming, it can just pretty much makes it the body just look different every time you see it. Every time it has a different shape, a different projection, that type of thing. I want you to look at my screen. Look at the screen now. I'm moving the cursor. All right. So here, look at this. This area here could just come right back up in and just form in a big circle. You know. Next time you see this thing, it just looks totally different. Now look, this is very important though. I want us to look at this. You remember just now we were looking at the bacteria and we said that the nucleus was not bounded by a membrane and that made it prokaryote. Now this is a unicellular organism, the amoeba. However, it is a eukaryotic organism. It is eukaryotic because it has a nucleus that is bounded by a membrane. That is bounded by, let me hear that again, a membrane. Right, by a membrane. Let's move on. All right, so we have what is called a contractile vacuole, and the contractile vacuole, it excretes water and waste material. I want you to observe what I'm doing on my screen here. All right, I want to point out some stuff here. Remember that when we are labeling, I just have to touch this, just give me a bit. Forgive me. When we are labeling, we want to ensure that the font size is the same. So there should not be a capital letter here. The font size should be the same. So this little area here would be the label, and this area here would have been the annotation, right? So this is labeling with annotation. Now remember, this method, drawing, labeling, annotation, is also describing the organism. So when we come upon the word describe, this is one way we could describe, by drawing something and providing labeled annotation. Let's continue. I have to use the examples as they pop up in front of me. Pseudopodia, false leg. Here it is. False leg. So it goes along by cytoplasmic streaming. I don't know if you have ever seen water thrown away on the floor and the water is just moving across the floor. The water has no legs in it, but the water is just moving across the floor. So pretty much that is how the amoeba would be moving. It's just flowing across the surface, pretty much like some water being thrown away on the floor, right? And then it could just, you know, break up and make some legs, more like some tentacle or something like that, and then we call that little leg portion looking thing a pseudopodia. Of course it doesn't look like a human leg or any animal leg that you know, so don't even say, but I don't see any foot. Oh yes, it has. We call these things leg. And remember, we say pseudo, it is false, and definitely it doesn't look like yours. All right, food being engulfed by the pseudopodia. I tell you, this organism is marvelous. Now look at the area right here, and you'll see the pseudopodia. It just curves this area in, engulfs some food that it has, and then it's going to just come right around and then close back this area, right? And then it's going to engulf. What was the engulf? Or it's going to be digesting the food on the inside. So this is just going to be locked in and become something like this over here. I hope you're looking at my screen. Are you paying attention? Good, perfect time to look. So it's going to, this area here is going to be coming like this in a short while. Now let's move on. Now the food vacuole, of course, that is where the food is digested. All right, Let's move on down. And we have the cytoplasm. Remember that we are still trying to contrast, compare the similarities, of course, and the differences between the cells we mentioned before. Remember, at any time you can go backward in this video, at any time you can pause, take notes, and ensure that you understand clearly what we're doing. You could leave us a comment, or you could say to your teacher, I understand or don't understand. I have been in touch with a number of teachers, and I've been getting some feedback. Remember to tell your friends to like, share, subscribe, and ensure that you share this video with as many persons as you can. All right, the last thing here we're going to be looking at with the amoeba is the cell membrane. All right, it controls substances that are entering and leaving the cell. All right, so this organism has a cell membrane. Make sure that you are able to compare these organisms, and remember, I have given you a definition. I have provided explanation for comparing. When you are comparing, you are looking at similarities and differences. All right, that's the easiest way for me to put it right now. I can't think of another way to put it. We are just looking at things that are similar and those things that are different. Let's move on. Identify. You know, this is a word that you'll see on your exam paper from time to time, and because we are so accustomed, probably in some cases, to lower order question, what is, when with these questions pop up with these new words, for some students it poses a challenge. We want to ensure that that challenge is through the window. It is easy for everyone. So if you are watching this video, ensure that you have somebody else that's learning with you, or you're teaching somebody. So identify. What do we do when we identify? We establish or tell who or what someone, something, or place is. So you are identifying. I want you to identify the date that Christmas is celebrated. Identify. It's like naming the date. So it is December 25th if you are in our geographical space. December 25th, we are able to identify. We can, in my country, would say we could hit the nail on the head, spot on, bullseye. Identify your teacher. Who's your teacher? You can tell who is your teacher. Identify your friend. You can say who is your best friend. And it doesn't require any long explanation. Just get to the point. The December 25th, the 25th of December, and there you'd have your answer, and move on to the next question. Now here we go. In this question, it wants us to identify the smallest in size of the following organisms, and it gives us the bacteria, fungi, virus, protozoa. Yes, we have not looked at fungi. Yes, we have not looked at virus, but guess what? I know somebody wants to do some more reading. Not everyone wants everything on a platter. Some person wants to go a little more. So just do a little more reading or a little more watching, and I'm sure you'll come up with this answer. Identify. Now, distinguish. This is another big word that appears on the exam paper. To identify differences between people, place, or thing. People, place, or thing. We want to identify. Distinguish. What makes a difference? What are your examples? I'm sure you can tell the difference between something, a fork and a spoon, maybe. Your shoes and your parents' shoes, maybe. A boy and a girl, just maybe. A car and a van, just maybe. And the list go on and on and on. Now look at example, the word example. It is a thing of its kind. So if you are going to be talking about examples, give me an example. You are talking about something that is of the same kind that you are talking about, that's there. So I'm asking now, an example of a mango. What would an example of a mango be? I don't know where you're from and what the common names are. We're talking about Mangifera indica. So an example of the Mangifera indica, an example of, and the syllabus does not require you to know botanical names. Mango, we have black mango in Jamaica, we have number 11 in Jamaica, we have East Indian in Jamaica, we have Julie in Jamaica, and I tell you, the list could go on and on, and we could just make a big mango song, right? But when we are talking about example, it is a thing of the same kind. So you would agree with me that all mangoes are pretty much of the same kind. Of course, they are going to look a little different, they are going to taste a little different and all of that. But once you see a mango, you can identify a mango versus an orange. Let's move on. So here we want to distinguish between prokaryotes and eukaryotes. Prokaryotic organism and eukaryotic organism. And you're going to be getting 2 marks. Now, I want you to look at this in an exam. The marks that is on the paper and the number of lines give you a clear indication of a value, how much you are supposed to write. So once you've mastered distinguish and you are looking at the weighting, don't go writing all the exam paper in 4 lines. All right. For this video, from this video, I want you to give example of each. So in the video, you would have been able to identify organisms that are prokaryotic and eukaryotic. I want you to give one example of each here. Now, using 2 examples show differences between plants and bacterium. I want you to show me difference between a plant cell and a bacterium. What's the difference? Now go back to the video, go through the video, look at both cells. I'm going to be tying the other video for cells in this video, so you should be able to make the difference, or to say what makes the difference. Identify 2 good uses of bacteria. Really? Is there anything good about bacteria? Really? You have good things to say? Oh, cheese? Sorry, I'm not helping you to answer the question. Just go back to the video, go back through the video, and how many times ever you wish, and ensure that you get full marks for these questions. Name. What does it mean? Name. To correctly identify people, place, or thing. Name. This doesn't require any long explanation. Name your school. My school is the cover page I. That's it. Name, or you could state, usually means to give. When using a question, in most cases, it means give. Alright? So name a bacteria, name a bacterium, and state its economical importance. Bacterium. Remember, I asked you to look at bacteria and bacterium. So you probably want to look at this question and leave something in the comments. Question number 6 to say if it's correct. But of course, the outcome is true. So you want to state the economical importance of bacteria. You want to name a bacteria, a bacterium, and you want to state its economical importance. And this is just the question for you to say which is correct. Here in this question, would it be bacteria or bacterium? Suggest to put forward a thought that is supported by scientific facts. So just don't go about and suggesting any old thing. This is what it is because I think that that is what it is, or I saw it in a cartoon, or I heard some men on the street. No. When you are suggesting, it should be rooted in facts. Alright? Good. So suggest the economical importance of knowing about microbes. Why do we need to know about microbes? Why? All right? Try that for me. Compare the movement of the bacteria to the amoeba. So remember cytoplasmic streaming versus the flagellum. Remember that, and tell me what's happening there. Now, using diagrams, compare the plant cell, animal cell, amoeba, and bacteria. So that's actually one letter size paper, and you're going to be separating it into 4 sections, and then you are going to draw all 4 annotated, of course, to show the differences and similarities as we are comparing. All right, I'm sure that you have it all pat, and remember, this is not an exam. All right, so you are going to go through the video again and again and again to ensure that you have mastered all these skills. The last activity here, I want you to make a list of the words explained and see how many of them your friends actually know. All right, how many of them they really can explain to you, how many of them they can really use or interpret were they to see it in a exam. All right, moving on down. So we are at the end of today's lesson. In our next class, we're going to be looking at cell specialization, or your teacher might say cell differentiation. So in our next lesson, we are going to be finalizing or wrapping up cells to move on to a new topic. You should have the cells video out by Tuesday thereabout. I'm sure that it should be out then, but until then, I want you to answer these questions, keep watching my videos, send me a comment, like, share, subscribe, get all your friends and teachers to subscribe to my page, because I also have some technology video for this distant learning. I've been doing technology videos for distant learning, and there might be some tech tip skills that you might find there. Let me know what you think. Leave me a comment. Thanks for watching the cover page. Later, remember to study.